Hello! We are UC San Diego's 2018 Senior Design Team 17. We have partnered with the J. Craig Venter Institute in La Jolla, California to present to you our latest research on the minimal cell. My name is Adam May, and my team members are JingYao Chen, Genghao Chen, Xiao, and Samuel Park. Your classmate, your pet dog, your parent's citrus tree. What do all of these have in common? They are all living. But what is it exactly that distinguishes life from non-life? And how does the genetic information inside all of us keep us alive? Researchers at the J. Craig Venter Institute began the quest to answer these questions nearly 20 years ago. Their goal was as clear as it was ambitious: to synthesize life.
We're here today to announce the first synthetic cell, a cell made by starting with the digital code in the computer, building the chromosome from 4 bottles of chemicals, assembling that chromosome in yeast, transplanting it into a recipient bacterial cell, and transforming that cell into a new bacterial species, to see if we could synthesize the bacterial chromosome so we could actually vary the gene content to understand the essential genes for life.
After more work, the developers of the synthetic cell reduced the size of its genome to only 473 genes. This new cell, a minimal synthetic cell, was given the title JCVI Syn 3.0, hereafter referred to as the minimal cell. However, 149 genes in the minimal cell remain functionally unclassified. This is a problem that our project is seeking to address. So, how do we determine the function of a gene? Traditionally, gene function is determined by performing gene knockout experiments, in which a single gene is removed from the genome and the resulting cellular phenotype is compared to that of the cell with the gene included. However, this approach is incompatible with the minimal cell. Because each gene in the minimal cell is essential for it to live, if any of its genes is removed, the cell simply dies. This presented us with a methodological problem. How do we study the genes in the minimal cell without killing the minimal cell? Our solution is to use CRISPR interference, also known as CRISPR-I. CRISPR-I has revolutionized functional genomics, enabling targeted and inducible inhibition of gene expression. Applying this technique in parallel would allow us to study the function of every gene in the minimal cell. The project consists of 4 main steps. First, we synthesize a plasmid library, in which each plasmid contains the gene encoding dCas9, as well as a guide RNA targeting a particular gene in the minimal cell genome. This process consists of multiple restriction digests, PCR amplifications, and E. coli transformations. This plasmid library will serve as the molecular instructions for our minimal cell. Second, we develop a library of minimal cells, each containing a single set of instructions from our plasmid library. Third, we induce CRISPR-I. Fourth, we take a snapshot of the transcriptome using RNA sequencing. And finally, we analyze the sequencing data to infer gene function. We successfully completed the first step of this protocol and are currently working to optimize the second step. We could not have done this project on our own. We will be forever grateful for the supportive leadership of Dr. Suzuki at the J. Craig Venter Institute, as well as the mentorship provided by former senior design student and UCSD alum Emanuel Vasquez. This concludes our video on the minimal cell. If you would like to learn more about our project, please visit our website at minimalgenomics.weebly.com. Thank you for listening.